Hi everyone, and welcome to the first lecture for the urinary system. So the urinary system is a lot of physiology, and for that reason it is split into 3 lectures instead of 1. It is the major excretory system in the body, and so we have 2 kidneys that produce excretory products, and they are carried by 2 ureters, which are tubes that essentially carry the urine from the kidneys where they're produced to the urinary bladder where it is stored. Now the bladder is emptied via an orifice called the urethra. So essentially, kidneys are taking in blood and they're filtering a large amount of blood to remove wastes from it. And that is what urine is. So blood goes into the kidneys where it is filtered in a process that we're going to see a little bit later by little units called nephrons. And they're filtering this blood in a multi-step process, and that is how urine forms. So essentially, urine is excess water, excess ions, and metabolic wastes, which are produced by breakdown of food and medications and just normal physiology as well. So let's quickly review the functions of the urinary system. I apologize for how wordy this slide is, but since a textbook is not required for your course, I just wanted to have as much information that I can fit into these slides. So excretion of wastes from blood is the most major function of the kidney. Regulation of blood volume and blood pressure. So here is a new concept. So the total amount of blood that is in circulation in your body is your entire blood volume, right? So blood volume and blood pressure are interrelated. So an increase in blood volume means that there's more blood to circulate in your body, and hence there's more pressure being exerted on your blood capillaries by this fluid. Now, why are we discussing this in the urinary system chapter? Because the more you pee out, the lesser your blood volume gets, assuming you're not ingesting any water. So producing more urine and not replenishing it can cause blood volume to go down, and when blood volume goes down, blood pressure goes down. So blood volume and blood pressure have somewhat of a directly proportional relationship. It also regulates blood solute concentration. So we've been seeing these ions all the way throughout ANP1 and ANP2. So in your muscles and nerves chapter, we looked at sodium ions, chloride ions, potassium and calcium ions. In your digestive system is where we started looking at HCO3 minus, which is your bicarbonate ion, and also your phosphate ions, which are part of your nucleic acids, because it's deoxyribose nucleic acids has a phosphate backbone. They are also part of your blood buffering system, which is to regulate hydrogen ion concentrations. And they also, if you recall our skeletal unit back in 241, we talked about bone calcium and blood calcium levels. So we talked about how blood calcium levels have a homeostasis, and a decrease of blood calcium levels can cause hypocalcemia, and excess blood calcium levels can cause hypercalcemia. So regulation of blood calcium is also done by the kidneys. So say you have temporarily excess calcium levels, so the kidneys are going to cause the excess calcium to be peed out. And if you have less blood calcium, or if you're calcium deficient, the kidneys are going to reabsorb that right into your bloodstream. So in terms of the overview of anatomy of the urinary system, there are two kidneys that are contralateral to each other. Here is your right kidney, here is the left kidney. The left kidney is slightly superior in position, whereas the right kidney is slightly inferior. Now, these are retroperitoneal, meaning that your entire digestive system and, the parts of the digestive system that are in your abdominal cavity and in your peritoneum are going to be anterior to what you're seeing right here. Now, this, on the medial surface of each kidney, originates your ureter, which is a tube that connects each kidney to a muscular sac, which is where urine is going to be stored. And this is the urinary bladder. Now, urine is stored in the urinary bladder, and it is voided through an opening called the urethra. We're going to look at the differences between a male and female urethra, the histology of these organs here in the later part of this lecture. So we just went over this, so I'm going to go over the slide and go right into the next one. So in terms of body positioning, you are seeing your liver right here in your upper right quadrant, and this is your right kidney. This is the left kidney. You are also seeing sort of a conical gland that sits on top of each kidney. They are known as your adrenal glands, also sometimes called the suprarenal glands, and they are parts of your endocrine system. So on the medial surface of each kidney, you are seeing, so this right here, the blue tube is your inferior vena cava. This red tube is your abdominal aorta right here. And you see branches of an artery and veins that are entering and exiting the kidney right here at this medial surface. And this point of inlet outlet on the medial surface is called the hilum. The other tube, the third tube that is also exiting out of the hilum, are your ureters right here. So the ureters are going through your pelvic brim and entering the urinary bladder on the posterior surface. Now this diagram kind of makes it look like they're attaching on the superior surface of the urinary bladder, but they're actually going behind and attaching sort of midway on the posterior surface. Now the urethra is the opening through which urine is voided. Now in image B over here, you are seeing a transverse section of the body and superior view. So we're looking at the body top down. And this space right here is your peritoneal cavity, which is where your stomach, pancreas, your small intestine, and large intestine is going to sit. You are looking at the cross section of both the kidneys here as well, and part of the renal artery, which is the red tube here, and the renal vein, which is the blue tube. The renal artery is a branch of your abdominal aorta, and the renal veins are going and joining your inferior vena cava. So I'm going to explain this content on the slide that follows this one. And so as you're watching the video, I strongly recommend that you fill out these blanks so that you are kind of making notes essentially for your test. And I also want to reiterate here that anything that is in bold is important information for your exams. So looking at the anatomy of each kidney, this is a frontal cut anterior view of the kidney. So the kidney is obviously a kidney, the name kidney bean comes after this. They are it is a convex, this is the lateral surface here, which is convex, and the medial surface is concave. Now the entire kidney is covered by a fibrous protective layer called the renal capsule. So it appears on a kidney, it appears sort of similar to what fascia looks like on your muscle. So it's a protective connective tissue layer. Now, once the kidney is cut, you're noticing these pyramid-shaped tissue bundles right here. So these structures right here are called renal pyramids. So the first thing you're going to notice is the renal pyramids. Now this outer area of the kidney, which I'm drawing on right now, the more superficial area of the kidney is called the renal cortex. And the deeper tissue, which is where the pyramids are, is called the renal medulla. So renal medulla and pyramids is not interchangeable terms, but rather medulla is that region of the kidney where the pyramids are housed. So the renal medulla has pyramid-shaped formations of the tissue. Now you will notice that between each pyramid you have this portion right here. So the portion of the cortex that essentially lives between the pyramids is called the renal column. So renal columns are basically cortex tissue that kind of form partitions between each renal pyramid. Now you will notice that the pyramids are sort of shaped like inverted triangles, and the apex, so this is the base of the pyramid right here, and the apex of each pyramid is opening into a funnel-shaped structure that is drawn in yellow right here. So this funnel-shaped structure is called the calyx. And the part of the pyramid that actually sticks into this funnel-shaped structure is called the renal papilla. So in other words, the renal papilla sticks into the minor calyx. So the minor calyx is this little funnel right here. So minor calyces from all the pyramids merge to form a larger collecting area right here, which are called the major calyx. The major calyx becomes and merges rather into a even larger area called the renal pelvis. And the renal pelvis continues on to become the ureter, which is going to go and merge into the urinary bladder. So to recap, the entire kidney is superficially covered with a connective tissue protective layer called the renal capsule. The outer or the more superficial layer of tissue is called the renal cortex. The deeper tissue is called the renal medulla. Parts of the cortex tissue that live between each renal pyramid is called the renal column. And the pyramids are shaped like inverted triangles, and the apex of each pyramid is called the renal papilla. The renal papilla sticks into a funnel-shaped tube called the minor calyx. Minor calyces merge to form the major calyx or major calyces, which merge to form the renal pelvis that continues on to become the ureter. Now here, on your medial surface, you have this region from here to here, which is called the hilum. Now there are 3 tubes that are entering and exiting the hilum. The first one is your red tube. It further splits, and we're going to talk about what these splits are here in just a minute. But this red tube is your renal artery. It is a branch off of your abdominal aorta. This blue tube is exiting the kidney and attaching to your inferior vena cava. That is the renal vein. And the yellow tube is the renal pelvis and the ureter. Now there are some spaces here between the minor and the major calyces that you can see. Now those hollows are typically filled with some adipose tissue, and those spaces are called the renal sinus. So the previous image was illustrated in color, but this is what an actual cadaveric kidney looks like. There's not as much color in it. So you're not seeing the renal capsule as much. It is a fibrous structure. You can clearly see the demarcation between, here's the cortex, and this tissue right here is the medulla. You are also seeing the pyramid-shaped formation here, which is the renal pyramid, and this little apex right here is your renal papilla. Now this is part of that funnel right here that I'm drawing on, which is your minor calyx, which is opening up over here into the major calyx. And here again you have a major calyx. This larger space right here is the renal pelvis, continuing on to become the ureter. The space between, you can kind of see the difference in the contrast right here of the tissue. That is your renal column. Here you are seeing the hollow space, and there's usually adipose tissue here. Some of it has been cleared out in this image. That is the renal sinus. Over here, you are seeing this one is the renal artery, and this one is the renal vein. You are noticing the color difference. The renal vein has a slightly sort of purplish tint to it versus the artery does not. So this right here would be your hilum. Now within the kidney, there are individual functional units called nephrons. So a nephron is not a cell. It is rather a structure that is made up of several types of specialized cells, including epithelial cells and different types of cells that we're going to talk about. But it is a structural and functional filtration unit within the kidney. And there are billions of nephrons located within each kidney. So this is a simplified view of a nephron. And so the nephron is a structure that parts of the nephron live in the renal cortex. So this lighter colored tissue right here is the cortex. This darker pink right here is the renal medulla. So there are some parts of the nephron that live in the renal cortex and other parts that dip down deep into the renal medulla. So let's start with the proximal end of the nephron. You have a globular or a nodular shaped structure that I'm circling right here. That is called the glomerulus. The glomerulus is a nodular structure that you can even see with light microscopes, and it has a sort of a filtration network that looks a lot like this, like a sieve. So this is the glomerulus. Following the glomerulus, also located in your cortex, is this tube right here. This is called your proximal convoluted tubule. Following the proximal convoluted tubule, you have a dive that goes down into the renal medulla. So this entire apparatus from about here to here is called the nephron loop or loop of Henle. It kind of looks like a hairpin. It's a hairpin loop. Now you will notice that the direction of fluid flow here, and please notice I'm not calling it urine, but I'm calling it fluid. We'll talk about what that fluid is going to be this way. So essentially, blood is going to come into the glomerulus from the afferent arteriole. It is going to flow through the glomerulus right here, and a little bit of blood is going to exit the glomerulus via the efferent arteriole. The fluid that does get filtered through the sieve goes in this direction. It goes through the proximal convoluted tubule. It goes down here, and for that reason, this arm of your loop is called the descending limb of the nephron loop. The proximal end of the descending limb is thicker, and it is made up of simple cuboidal epithelium. The distal end of the descending limb is called the thin segment because it is made up of simple squamous epithelium. We will talk about the difference in physiology of these two areas in a little bit. Now this is where the hairpin bend happens, and that segment where the fluid is going this direction is called your ascending limb. Now in the ascending limb, the proximal segment is the thin segment, again made up of simple squamous epithelium, and the distal segment is your thick segment, which is made up of simple cuboidal epithelium. Now past that, this area here, or this segment, we're back into the renal cortex. So the only part of the nephron that actually dives into the medulla is the nephron loop. Now this segment is called the distal convoluted tubule. Now, several distal convoluted tubules from different nephrons come and merge into a common tube. Now, stuff that goes through this tube here, called the collecting duct, is now called urine. So, collecting ducts from several different pyramids join to form the minor calyx. The minor calyces join to form the major calyx, the renal pelvis, and from there the ureter. Now in that diagram that I mentioned, the glomerulus and the capsule that's covering it is called the renal corpuscle, and we're going to talk about what individual components go in the renal corpuscle. Then you have your proximal convoluted tubule, and your corpuscle and the proximal convoluted tubule are both located in the renal cortex. Next, you have the nephron loop, which consists of the descending limb followed by the loop and the ascending limb, and both the descending and ascending limb have segments of thin and thick respectively. And this is what dives down into the medulla, followed by the distal convoluted tubule, which is also located in the renal cortex. So this is an illustrated and highly zoomed in version of different types of nephrons. So this lighter pink tissue, that is your renal cortex. The darker pink tissue is your medulla. This apex that you're seeing right here, it is your renal papilla. And right here, you have your minor calyx. So there are nephrons that are located farther superficial. into the cortex, and there are nephrons that are located with, and those nephrons that are farther, that are more superficial into the cortex are called cortical nephrons. The nephrons that are located closer to the junction of the cortex and medulla are called juxtamedullary nephrons. So the loops of Henle of the cortical nephrons don't go as deep into the medulla, but the loops of the juxtamedullary nephrons go a lot more deeper into the medulla. And you can see that there are collecting ducts that come and attach to, there are distal convoluted tubules of several nephrons that come and attach to collecting ducts that open into the minor calyces. So going into detail about each segment of the nephron here, starting with the renal corpuscle. So right here is renal corpuscle, or spelled right here. So the renal corpuscle consists of a horseshoe-shaped structure, this right here, called the Bowman's capsule or glomerular capsule. Now the Bowman's capsule is engulfing a sieve-like structure that you see right here as a web of blood capillaries. That is your glomerulus. Now here is your afferent arteriole, which is how blood is going to come into the glomerulus. And here is your efferent arteriole, through which blood is going to exit the glomerulus. Now, how you tell the difference between the afferent and efferent arterioles is by their diameters. The afferent arteriole is going to have a larger diameter, and the efferent arteriole is going to have a smaller diameter. So when we start talking about nephron physiology, which will be in a separate lecture PowerPoint, we're going to talk about why the afferent arteriole has a slightly larger diameter than your efferent arteriole. Now, as blood comes into your glomerulus, parts of blood are going to be caught here by this sieve. And there are going to be parts of blood, the more liquid parts, that are going to get squeezed out here in this space of the Bowman's capsule. So this capsule is essentially lined with simple squamous epithelium. Now the glomerulus itself has specialized cells called podocytes that are not visible here, but we're going to talk about them in just a little bit. So glomerulus right here is a network of blood capillaries, and I've already talked about the difference between the afferent and efferent arterioles. So take a look right here. The one with the wider diameter is your afferent arteriole. Blood enters through here, goes through this tube, and flows all the way in this sieve of your glomerulus. Here is your Bowman's capsule, and the Bowman's capsule, you can see here, is lined by simple squamous epithelium. This hollow area is called the capsular space. Now over here, you see these amoeba-shaped cells with a nice nucleus right here. Those cells are called podocytes. So the parietal layer of your glomerular capsule, also known as the Bowman's capsule, is lined with simple squamous epithelium. The visceral layer is lined with cells called podocytes. Now your capsule, as well as the glomerulus itself, goes by the name renal corpuscle. This is your efferent arteriole. Now the efferent arteriole has a smaller diameter, and this is how blood is going to exit the nephron. Now this is where your proximal convoluted tubule begins. There are a few other cells that I'd like to talk about right here that are wedged. So this is your afferent arteriole. This is going to be your proximal convoluted tubule. So this is a very, I'm not the best artist here, so bear with me. So this is going to become the loop of Henle. It's going to go deep down into the cortex, down the cortex and into the medulla. And then it's going to come back up. You're going to have your distal convoluted tubule right over here. So imagine the nephron kind of shaped like that. So there are specialized cells called juxtaglomerular cells. So juxtaglomerular cells are a type of specialized cell that are located, they're wedged essentially in the space between your afferent arteriole and your distal convoluted tubule. So I'm going to change the color of my pen here and do it in green. So the cells that I'm shading here right now, those are your juxtaglomerular cells. And when we start talking nephron physiology, we are going to talk more about what the function of these cells are. So those right there are your juxtaglomerular cells. Now, in the lumen of your distal convoluted tubule itself, you have specialized cells that are rather densely packed. I'm drawing on them right now. Now, these cells that are inside your distal convoluted tubule here, they are called your macula densa cells. And together, these juxtaglomerular cells and the macula densa cells make an apparatus that plays a role in whether your urine is concentrated or whether it is dilute. So they play a role in reabsorption or secretion of different solutes within the urine. So these two types of cells together and the afferent arteriole is called the juxtaglomerular or the JG apparatus. So the renal corpuscle is actually fairly porous. So between the podocytes, so we saw those amoeba-like cells, the gaps between those podocytes are called fenestration slits or fenestrae. And the glomerulus is like a sieve, right? So it's catching all your blood cells. There shouldn't be red blood cells and white blood cells in urine, ideally. So your glomerulus, which is the sieve, is supposed to catch your blood cells, any kind of large blood proteins like albumin and globulin is supposed to be caught by the glomerulus and exits through that efferent arteriole. The porous visceral layer allows, so this fluid is coming in through your afferent arteriole and it is being squeezed out between your fenestration slits and ends up in your capsular space. And we did discuss the difference in the diameter of the afferent and efferent arterioles. So the whole glomerular capillary network within the glomerulus has higher pressure because the efferent arteriole is smaller or has a smaller diameter than the afferent arteriole. So we did talk about the juxtaglomerular apparatus, which consists of two components, the juxtaglomerular cells that are wedged between your afferent arteriole and your distal convoluted tubule. They're wedged between that space, and your macula densa cells are located in your distal convoluted tubule itself. Now this slide, put a star mark on the slide, and in lab we will be looking at a lot of these. We'll be teasing out the histology of this a little bit more, but this image shows you the histology of every portion of the nephron. So let's start right here at the renal corpuscle. So let me change the color and maybe make it green so it's a higher contrast. So right here you are seeing the glomerulus itself, and the nuclei that you are seeing there are the nuclei of the podocytes. This space right here, the hollow space, which is sort of the a mirror image of a C shape or a horseshoe type shape, that is your glomerular capsule or the Bowman's capsule, and the space is the capsular space. And you can see the Bowman's capsule is lined by simple squamous epithelium, and the glomerulus has your podocytes. Next, you have your proximal convoluted tubule. So the proximal convoluted tubule histology is right over here. You can clearly see the lumen. Now it is lined by microvilliated, simple cuboidal epithelium. You can see the microvilli here fairly clearly, and the job of the microvilli here, much like the digestive system, is to increase surface area of the cell. And you have tight junctions, which is a reference back to the first unit of ANP2. You have tight junctions between these cells so that the fluid that's flowing through this tube is not leaking out of the nephrons. So they are impermeable or waterproof junctions. From your proximal convoluted tubule, we are going to go into the descending limb of the loop of Henle right here. So the descending limb, the proximal segment here, is the thick segment, followed by this over here is your thin segment. And the thick segment has simple cuboidal epithelium, and the thin segment has simple squamous epithelium. Now we're back into the, this was the descending limb. We're now going into the ascending limb. Right here. And the ascending limb first has your thin segment, followed by thick segment. So you are seeing right here the descending limb histology. And over here, we're not seeing a separate ascending limb histology here. But right here is the collecting duct, right? Several nephrons share a collecting duct, and the collecting duct has simple cuboidal epithelium as well. And you can see the lumens of those right there. So that was our anatomy and histology of the filtration units of your kidneys, which were the nephrons. The next part, we're going to end this PowerPoint by going over the vasculature in the kidneys and talking a little bit about the lower urinary system, which is your ureters, bladder, and urethra. So let's start with the arteries of the kidneys, which right now we're looking at the red tubes. So here you can see the hilum right here, and you're going to have your abdominal aorta, which is going to branch off and become the renal artery. So we're going to start right here at the renal artery. The renal artery is going to go in through the hilum, and it is going to split into segmental arteries. So these two right here are segmental arteries. So segmental arteries are further going to split, and they're going to go down the sides of the pyramid. So this segment right here is your interlobar artery. So if it goes down the side of the pyramid right here, it's interlobar artery. Then it curves or it arcs around the base of the pyramid right here. That segment right there is called the arcuate artery. From there, it splits into smaller blood capillaries that radiate outward into the cortex. And so those are called the cortico-radiate arteries. They radiate out into the cortex. So the arterial system here is, you have your abdominal aorta that splits into renal arteries. Renal arteries split into segmental arteries. Segmental arteries go down the sides of the pyramids, and those are the interlobar arteries. They curve around the base of the pyramid, called arcuate arteries, and then they radiate out into the cortex, and those are your cortico-radiate arteries. From there, they are going to become smaller blood capillaries, and they are going to be your afferent arterioles that carry blood into the nephron itself. So here you are seeing your cortical radiate artery, which is what we saw on the previous slide. It's going to become a smaller blood vessel, your afferent arteriole. So from here, blood flows through the glomerulus and exits out through your efferent arteriole that I'm shading on right now. So the efferent arteriole further splits into a network or a web of smaller blood capillaries. Now these smaller blood capillaries, if you notice them, they are forming sort of a mesh around your proximal and distal convoluted tubules. So those are called your peritubular capillaries. So peritubular capillaries are the blood capillaries that live around your PCT and your DCT. Now, right here is where your loop of Henle is. So as you go down into the medulla, the blood capillaries here, again, they're forming a web. Now, as the fluid is going through the descending limb and going up the ascending limb, you are noticing these blue tubes. So the blue tubes are little venules or deoxygenated blood capillaries. So this mesh of blood capillaries that lives around your nephron loop in particular is called the vasa recta. So vasa recta is a web of blood capillaries surrounding your descending and ascending loop of the loop of Henle. So as you notice, the blue systems right here are going in, and they are forming a tube called the cortico-radiate veins. So we're going to have similar veins to the arteries we just saw, and we're going to start backwards with the cortical radiate veins and come out towards the hilum. So right here, we're going to start with the cortico-radiate veins, which are the blue tubes that you see in the cortex. They are going to come and form your arcuate vein, which is at the base or curved around each pyramid. So from there, you are going to see the interlobar vein, which is on the side of each pyramid, right there. From there, different interlobar veins merge into the renal vein, which then exits the hilus and goes and merges into the inferior vena cava. So if I were you, I would sort of draw a flow chart side by side of the arterial and the venous system. So we're going to do this also interactively in the lab when we meet, but it helps to have this pathway kind of charted out before we kind of address this in lab. So the next component, so we've finished the kidney now, and the next component we are going to look at is the ureter. So the ureter is essentially a tube that carries urine from point A, which is the kidney, to point B, which is the urinary bladder. So the ureter is a tube. So what you're looking at is the cross section, and in here you will notice the lumen. So the lumen is highly folded. It has several pleats, if you will, in it. And this is a tube, so we're going to see all the walls of the tube, the same kind of walls that we saw in the digestive system. So you are going to have the mucosa, which includes your epithelium, the lamina propria, and your muscularis mucosae. Then you're going to see the submucosa, and then you're going to see the muscularis externa, which has your circular and longitudinal muscles, which are smooth muscles, followed by adventitia here in the beige. Now, this being the lumen, now this tube has to, the fluid that flows through here is slightly acidic, and these cells are commonly shed a little bit. So the epithelium right here is type of a stratified epithelium. You already know this. This is the transitional epithelium. And here you see the basement membrane of the transitional epithelium, followed by the lamina propria, which is going to have areolar connective tissue. Here's a light micrograph. So this darker violet that you see right here, that's your transitional epithelium. You are seeing the, right here, that is your lamina propria, and you're going to have your muscularis mucosae. This area right here, you're probably seeing the submucosa, then your inner circular and your outer longitudinal muscles that are part of your muscularis externa. Now the urinary bladder is located posterior to the pubic symphysis right here, and the ureter joins the posterior surface. So the ureter does not open up on the top here, but instead it goes in the back like that, and the opening is actually right there. So the ureter goes on the posterior surface, or the dorsal surface, and opens up, and this area right here between the two urethral openings and your internal urethral sphincter is called, so these are imaginary lines here. This area right here between the two urethral openings and your internal urinary sphincter or urethral sphincter is called the trigone. Now, your urinary bladder is made up of a muscle called the detrusor muscle. So it is a smooth muscle. Here's the peritoneum. The bladder is located outside the peritoneum. And the urinary bladder on the inside has a lot of pleats, just like your stomach did. So these pleats that are inside of the urinary bladder are again called rugae. And here is your internal urinary sphincter. This is the urethra. Right here is your external urethral sphincter and your urethral orifice right there. So the histology is again transitional epithelium. This is the lumen of the bladder. These are your mucosal folds, also known as rugae. And you have your transitional epithelium, your lamina propria, made up of areolar connective tissue. Now, there is a bit of difference between the male and female urethra, and we're going to go over that right now. So here is your urethral openings. Here's the trigone, and the trigone sort of forms a funnel that opens into the internal urethral sphincter. So the female urethra is a lot shorter than the male urethra, and for that reason, females are more susceptible to urinary tract infections. So right here, you are going to have your internal urethral sphincter, which is smooth muscle. This is a urethra itself, which is a lot shorter, and here is your external urethral sphincter, followed by the urethral orifice through which urine exits the system. Now, throw back to your muscles unit. Remember the bulbospongiosis muscle that we talked about, and the ischiocavernosus muscle in the pelvic floor. So the urethra on the lateral sides is flanked by your bulbospongiosis muscle. The male urethra is a lot longer than the female urethra, and there are parts of the male urethra that are shared by the reproductive system. So we'll start here at the urethral openings. This is the bladder made up of detrusor muscles. You have your trigone, which is where the internal urethral sphincter is, right here. Then urine is going to flow through this segment. The blue segment of the male urethra is called the prostatic urethra because it goes in the middle of a gland called the prostate gland. So this right here is your prostate gland, and the prostatic urethra goes through the prostate gland. Now, this is the bulbospongiosis muscle right here. And so the short green segment that goes through those muscles, which is where the external urethral sphincter is called the membranous urethra. And then you have the base of the penis, and this purple segment of the male urethra, which is also its longest segment, is called the spongy or the penile urethra. And the external urethral orifice is in the glans or the tip of the penis, through which urine exits the system. So I'm going to stop here, and in the next PowerPoint, we're going to start looking at the nephron physiology.